Hi, this is Mr. Wartsky, and today we are going to start our first screencast on the cell cycle or mitosis. And once again, if you have any questions about this, you can go over the video again. You can read your textbook or maybe even ask me in class. Remember, you always also pause when you're doing diagrams, and you will be drawing a lot of diagrams in this unit. So, we know that in you people, there are 46 chromosomes per cell, except for gametes, and that's the next topic, but it's okay. So there are 46 chromosomes per cell. Each cell has 46 chromosomes, and each cell will get 46 chromosomes when the cell divides. So let's define some terms. First, somatic cell is a body cell. Basically, it is any cell except for a gamete. A gamete is a sex or reproductive cell, and there are only two gametes, sperm or egg. The genome is the cell's total DNA allotment, and those are the 46 chromosomes. And a chromosome is one long strand of DNA which is wrapped around histone proteins. On the DNA, you have many different genes or sections of DNA that code for proteins. And a chromatid, this is new, is a copy of a chromosome. So a chromatid is a copy of a chromosome. Usually the cell starts off with each chromosome being one chromatid. So in the next we have a picture which you don't have to draw. We see a picture of a chromosome. Right? Now we didn't talk about the centromere, which is the region where the spindle fibers attach, as well as the place where the chromatids attach. We have one chromatid here, one chromatid. Each is an exact copy of each other. How do you make chromatids? DNA synthesis, but that's a story for later in the broadcast. You will notice that there's a kinetic core, and not many people talk about the kinetic core. The kinetic core is where the place where the chromosomes attach. This is the centromere region. This structure is the kinetochore. Spindle fibers will attach at the kinetochore, and then those will move the chromatids around. So the kinetochore is where the spindle fibers attach. That is found in the centromere region where the two chromatids are attached. The cell cycle is broken down into 4 phases. Now it's a cycle, so it goes in a circle. We're going to start off with G1 because that's a good place to start. G1 stands for gap 1, and then gap 1 the cell grows. So how does the cell grow? It goes through protein synthesis. And you know all the steps to protein synthesis, so you should be able to describe G1 in great detail. S stands for synthesis, specifically DNA synthesis. This is where they go from one chromosome is made up of one chromatid to a one chromosome is now made up of 2 chromatids. And since you know everything there is to know about DNA synthesis, you can describe the S phase completely. G2 is a growth and protein synthesis. We also see the mitotic spindle is formed. Now, the only thing that you don't know about is the mitotic spindle, but you will. And M is mitosis, where the nucleus divides. Nucleus divides in mitosis, and the cell divides in cytokinesis. So it's mitosis and cytokinesis. And here is the cell cycle. We have G1, S, G2, and M. It's a cycle. So some people might have questions about how long these things stay in the cell cycle, different places of the cell cycle. We have G1, where it stands for anywhere from 6 to 12 hours, S, 6 to 8 hours, G2, we go from 3 to 4 hours, and M, which is approximately 1 hour. Now, GO is a resting phase, and we'll talk about this in a second, where the cell can actually exit the cell cycle and spend their time in GO and not divide at all, which is fine. So there are 3 checkpoints in the cell cycle, 3 places where the cell cycle can stop. The first G1 checkpoint is in G1. That's called the restriction point, where when it goes past this restriction point, or the R point, the cell will commit to the cell cycle. So if it's ready to go into S, it will just go right into S. Now, if it's not ready to go to S, it will enter something known as GO, and it will wait and wait. The protein cyclin-dependent kinase, or CDK, when that increases in the G1 stage, then the cell will start and commit to the cell cycle. The second checkpoint is between G2 and M. So the cell looks at the DNA. If the DNA after the S phase is okay, then it is ready for M. If the DNA is not okay, it will not go through the cell cycle and it will stop before M. Now there's a third checkpoint, and we're going to talk about that when we get into mitosis. Mitosis is the duplication and division of the nucleus. Cytokinesis is the division of the cytoplasm. Please don't get the two confused. In mitosis, there are 5 phases. You learned about 4 when you were in 9th or 10th grade, but now there are actually 5. And they are prophase, prometaphase, metaphase, anaphase, and telophase. Now, interphase prepares for mitosis, and that's your G1S and G2. Altogether, it is IPP, MAT, or IPMAT, and that is the cell cycle, where you have interphase, which is G1S, G2, prophase, prometaphase, metaphase, anaphase, and telophase. You didn't have prometaphase when you took biology the first time. So my job is to go through the different phases of the cell cycle and for you to draw the pictures down. So the first phase is interphase. And interphase is G1S, G2. Now you should be able to describe protein and DNA synthesis with great detail, and I'm sure you will have to do it shortly. So you should know interphase. Now you're going to draw a picture of the cell here, and this is the nucleus, and that's the nucleolus, and you're going to label it interphase. So do that now. If you need to, hit pause. In prophase, chromosomes condense. So there's a protein called M-phase promoting factor, or MPF, and it is a kinase. What this kinase will do is it will add phosphates to the chromatin, and the chromosomes will condense. As the chromosomes condense, the nucleolus will disappear because the chromosomal loops are pulled out of the nucleolus. If they're pulled out as the chromosomes condensing, the nucleolus, or the area of high concentration of chromatin, will disappear, so the nucleolus disappears. In this part, the chromosomes are made up of 2 sister chromatids because of the S phase, where the S phase produced through DNA synthesis a second copy of the DNA strand. Here is a picture of prophase. Now you'll notice that these right here, these are called centrosomes. 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 There are 2 centrosomes, and they will migrate to the opposite poles. These centrosomes produce spindle fibers. Now that is not written anywhere, so you're going to have to write that down from my voice. Centrosomes, they divided. If you take a look over here, there's only 1 centrosome, but here there are 2 centrosomes. So the centrosome divided, they'll migrate to the opposite poles, and the centrosomes produce spindle fibers. Spindle fibers. In prometaphase, the nuclear membrane fragments. How? Well, M phase, and the promoting factor that we talked about before, will also add phosphates to the nuclear membrane, and it will break up. The spindle fibers from the centrosome will connect to the chromosomes. And you're asking yourself, spindle fibers? Yeah, spindle fibers. Spindle fibers are made up of actin and tubulin from the cytoskeleton. So the cell actually disassembles the cytoskeleton with the actin and tubulin, takes it away. The cell becomes more flexible so it can divide. As it takes away the actin and tubulin, the centrosome organizes those 2 proteins into spindle fibers. Now the centrosomes, the centrioles are associated with centrosomes only in animal cells. The centrosomes are migrating to the opposite poles, right? Now the tubulin has a positive and negative end, and it only adds to the positive end. So that's how tubulin gets longer. As it gets shorter, it subtracts from the positive end, and the tubulin gets shorter, so it can move around the chromosomes. There are three types of spindle fibers. There's the kinetochore spindle fibers, and if you take a look at the diagram, they're here, and they attach from the centrosome to the kinetochore. Polar spindle fibers go from centrosome to centrosome, centrosome to centrosome, and these actually act as an anchor by keeping the centrosomes apart, whereas the kinetochore spindle fibers move the chromosomes around. The aster spindle fibers are here. They move from the centrosome to the cell membrane, again, centrosome to the cell membrane, and they also act as an anchor to the centrosome. So we have two anchors. We have the polar spindle fibers, and the aster spindle fibers are anchors, and the kinetochore spindle fibers will move the chromosomes around. This is a picture of prometaphase. Oh, what happened there? This is a picture of prometaphase. We have the centrosomes forming the spindle fibers. These spindle fibers will be aster, then we'll have some going from spindle fiber to spindle, centrosome to centrosome. This is polar spindle fibers here, and then some will start to connect to the chromosomes, and those will be the kinetochore spindle fibers. Also, please show somehow the nuclear membrane breaking up. You can just put, erase parts of it so that you know that the nucleus is breaking up because MPF is phosphorylizing it. We'll start one more phase here, which is metaphase. So the kinetochore spindle fibers will lengthen and shorten so that the kinetochores line up on the metaphase plate. Metaphase plate is like the equator of the cell. It's in the middle of the cell. This is where the third checkpoint is found. Okay. Now, what happens here is if the chromosomes are not lined up, the cell will not finish mitosis. It will stop the cell cycle right there. So the chromosomes have to be lined up in order for the cell to continue dividing, or the mitosis. If the chromosomes are lined up, then there is a protein called anaphase promoting complex, or APC, that is produced during that time. And here is a picture of metaphase when the chromosomes lined up. These are the aster spindle fibers here. We have the kinetochore spindle fibers there, and then we have the polar spindle fibers that go back and forth from centrosome to centrosome. Once again, the aster spindle fibers go from the centrosome to the cell membrane. The polar spindle fibers go from centrosome to centrosome, and the kinetochore spindle fibers will move from the centrosome to the kinetochore. And that will be the last slide for this screencast. Please listen to the second screencast in the series, and we will finish the cell cycle. Have a good night.